Now we'll summarize the whole thing that we have learned till now. What is important regarding enzyme nomenclature and mechanism of enzyme action. Now, as I said, enzymes are all proteins except the ribozymes. They are catalysts with high catalytic efficiency. Without an enzyme, the rate of reaction would have been 100 to 1000 times slower. So they are all proteins except ribozymes. That is important out here. Now enzymes are classified into 6 classes. This is also important because we are going to talk about any enzyme can be asked as belonging to which class. So please remember that it is in a particular order. Can you give an example for oxidoreductases? We have the example of all the dehydrogenases belong to the class oxidoreductase. Can you give an example for transferases? All the kinases belong to the class transferases. Can you give an example for hydrolases? All the digestive enzymes belong to the class hydrolases. Example for lyases. Lyases means they help in the removal of a bond. So all the decarboxylases and the synthases, the reverse reaction is also possible, come under the class lyases. Isomerases is easy, and ligases means they utilize ATP to form bonds, for example carboxylases. Now please remember that enzymes are highly specific. We have different types of specificity that is possible, like bond specificity, group specificity, absolute specificity. All these are the different types of specificities that are possible. Now again we go on to the mechanism of enzyme action. Please remember that a substrate has to form a complex with an enzyme, and the complex is formed at the site, at the active site, and this is an induced fit model. Now once it forms a complex, what happens next? The catalytic residues may act on it, and the binding residues also help in the formation of this enzyme substrate complex. Now the enzyme will stabilize the transition state, will help the substrate to reach the transition state faster by lowering the activation energy that is required, and this provides an alternate pathway by lowering the activation energy and stabilizing the transition state. Once it does that, what does it do? It brings about catalysis by proximity and orientation, by acid base, or by covalent catalysis. It brings about, or by stress and strain, it brings about catalysis. So once it does that catalysis, it results in the formation of release of the product. Please remember that in all this, the important thing as far as MCQs are concerned is that they do not alter the equilibrium constant of the reaction. That is important. Equilibrium constant, or K equilibrium of the reaction, is not altered. So if a substrate is supposed to reach to a product, the equilibrium between that, how much of substrate and how much of product is formed, that is not going to be changed by enzymes. Now the measurement of enzyme activity is basically by the international units and the SI units. So with this we come to the end of how enzymes act as what is the mechanism of enzyme action. We'll go further on this in the next session.